releasers. Now, another example of the visual releaser is an example of the digger wasp, Philanthus triangulum. Now, see, this wasp builds a nest burrow in the sand that contains developing larvae, which she supplies with food. We have seen earlier that this is an example of FFP. The digger wasp does not need to learn how to build a nest. Tinbanger used visual cues that how the digger wasp returns to the nest. So he conducted an experiment, and in the experiment what he did, when the digger wasp was in the burrow, Tinbanger placed a circle of pine cones around the entrance. If you see in the testing mode, the pine cones, they were placed around the net. This is the first stage. So when the digger wasp came out, it flew around the cones. So the cones, they were the visual clues. Nest, coming out, and now she revolves around. So when she emerged, the wasp reacted to the new situation by wavering orientation flight, training condition before flying off. So in the first stage, this is the first stage. Second stage, it's flying around the pine cones. So the digger wasp now identifies the nest through these pine cones. But what he did, Tinbergen, when the digger wasp left the nest, he shifted the pine cones. He shifted the pine cones away from the nest. Now you see this, these are the pine cones, these are shifted. So pine cones moved here, so the nest is not enclosed by it. But when the digger wasp returns, it returns to the pine cones, even though nest is not present there. So returning with prey, testing condition, she flew to the circle of pine cones, even though it had been moved during her absence. So we can see this is the nest, and the wasp returns to the circle of the pine cones. So these are some example of the visual cues. Now the auditory releasers. The auditory releasers are the SignStimulus auditory signal, like the humming sound of the male insects. See, we have seen the songbirds such as cuckoos, nightingales, starlings establish their territory and attract the female for mating by singing, often for several days. The song is a signature stimulus of individual bird, as it is specific. Birds of different areas sing in different dialect and elicit different response. Stridulatory organs of some insects, such as the cicadas and crickets, are strong sign stimuli during the breeding season. Co-cue calls given by the tree frogs, Eulithodactylus, are strong auditory releasers for males, as well as female frogs of the same species. The chemical releasers. The third type of the releasers are the chemical releasers. Chemical releasers, pheromones or the external hormones, these are different type of volatile chemicals released in the atmosphere that act as releasers on the individuals of the same species or other species. Pheromones affect the individuals of same species, while pheromones affect other species. Sex pheromones in insects are produced by the females and perceived by the enlarged antennae of males of the same species. sometimes from the distance of a couple of kilometers. In vertebrates, in some vertebrates, the sex pheromone copulin is secreted by the female in estrus. Alarm pheromones are produced by skin cells of several species of fishes that have schooling or shoaling behavior. Alarm pheromones are also released by the stinging honeybees and wasps, which attract other individuals of the hive to attack. Ants and termites produce trail pheromones from the posterior end of abdomen, which help them to follow each other. Sign stimuli bring about responsiveness in target individuals, which show consummatory behavior. The responsiveness diminishes as the consumption proceeds and the energy is released. Now see, another type of stimuli is the supernormal stimuli. We have seen this example of gull, and the tip of the beak has a red spot, which the young girls use it as a sign stimulus. So this is the accurate model, the red spot, and this is a pencil with a red spot. Now this pencil with a red spot acts as a supernormal stimulus. So what is a supernormal stimulus? The supernormal stimuli is a phrase used to describe artificial stimuli that are more effective than the real thing in eliciting a behavioral response. So it's a stimuli which is similar to the original stimuli, but it responds or it elicits a response which is much higher than the original behavioral response. So we have seen that the herring gulls chicks peck at a red spot on their parents' bill to induce their parents to regurgitate food. We have seen a video earlier also in the fixed action pattern. You can see that again in that lecture. Now chicks will also peck at a model consisting of a red spot against a yellow background. However, it is possible to construct a model that is even more effective than a real head by using a red pencil with 3 white bars at the end. Now see, a pencil with 3 white bars at the end is an example of supernormal stimuli. In this experiment, the supernormal stimulus received about 25% more pecks from the gull's chick than the natural head, a model of an adult head or a model of the adult's bill. See, this is the result. This is the pecking rate of a natural head. Standard model and only bill. And this is a stick with 3 bars, red pencil or a red stick with. If you see, the highest response is of this. So these are supernormal stimulus. That is the supernormal stimulus. They are artificial stimuli. They are not natural stimuli. They can be produced in the laboratory to elicit different responses. So we have seen today that there are 3 types. In this lecture we have seen 3 basic types of stimulus: visual stimulus, the auditory stimulus, and the chemical stimulus, and one type of artificial stimuli, that is the supernormal stimulus. So this completes the SignStimulus part 2. Thank you.